EMI504 superlab. We're identifying similarities and differences between models. Before you get started watching the video, you want to make sure that you have the EMI504 skill sheet in front of you. It should be the one with the circle drawn diagram near the bottom right. So genes or bacteria called plasmids. You also want to make sure you have something right. Of course, if at any point during the video you feel like you are confused, you should stop and rewind and watch it again. Today what we're going to be looking at is the EMI504. EMI504 is almost always assessed as part of the conflicting viewpoint passage of the Boolean scientists. In order to do this, we should have a basic understanding of each of the scientific viewpoints. In this case, they're called models. A model is a scientist's way of summarizing how a system works based on some evidence that they've collected. For each model, you should be able to explain what their hypothesis is, usually defined in the first sentence of their argument, and at least one major piece of evidence that they use to support that hypothesis. EMI504 asks you to be able to compare and contrast the hypotheses or ideas. Let's get started by taking a look at an overview of what these four scientists are going to be arguing about. At this point, you want to have an implement of writing in your hand, and if you come across a word that you are confused about, you should circle, highlight, or underline. Now, to this point, we haven't talked about genetics yet, so there's probably quite a few words here that will jump out and we'll get confused. We'll get through the vocab, so it won't look bad. Many bacteria contain plasmids, small circular DNA molecules. Plasmids can be transferred from one bacterium to another. For this to occur, the plasmid replicates, produces linear copies. The relative position of the genes is the same in the original plasmid and in the linear copy, except that the two ends of the linear copy do not immediately connect. While replication is occurring, one end of the linear copy leaves the donor bacteria and enters the recipient bacteria. Thus, the order in which the genes are replicated is the same as the order in which they are transferred. Unless this process is interrupted, the entire plasmid is transferred, and its two ends connect in the recipient bacteria. 4 students studied the way in which 6 genes, F, X, R, S, A, and G, on a specific plasmid were donated by a type of bacterium. That's what the figures show. The students determined that the entire plasmid is transferred in 90 minutes and that the rate of transfer is constant. They also determined that the genes are evenly spaced around the plasmid, so one gene is transferred every 15 minutes. They disagree, however, about the order in which the genes are replicated and transferred. 4 models are presented below. Before we get into the 4 models, let's start to act through some of this vocab stuff. Are there any words that you circled, highlighted, or underlined, words you were confused about, or you've never heard of before? How would you want? Recipients. Recipient bacteria. Second paragraph, second line. Oh, nice. Any other words that are getting in our way? We have a couple new vocab words. Plasmid and plasmid replication. Bacterium. It's a little different than our typical. Okay. And last call for vocab, going slides. Sold to plasmid. So remember, there's 3 ways that we can figure out any word in science. We can have it defined for us in the passage. We can use context clues or background knowledge, or we can look it up. We actually we can't have a dictionary, so we try to avoid the last one. Plasmids, that first word, is it defined for us in the passage? Next, what's the definition?
Small circular DNA molecules.
Nice. Small circular DNA molecules. Your DNA is not circular. Your DNA is in a long string. Like that. Right? Bacterial DNA is actually in a loop. Right? That's the plasmid that we're talking about there. It's a small circular DNA molecule. They can be transferred from one bacterium to another. Is bacterium defined for us? No, there's no definition. What word do you see in there that jumps out at you? It's like, hey, that looks pretty familiar. Or at least a root part of it. Bacteria. Yeah, you see some like bacteria. All right, it's pretty consistent, but it ends in an IUM. Why is that? This is actually the singular for the word bacteria. Bacteria is plural for bacterium. So whenever you say the word bacteria, you're talking about multiple of that. Right? So there's got to be at least more than one bacteria. And this to occur, the plasmid replicates. What do we mean by that word replicates? That was defined for us. What's that word replicates mean? Nice, makes a copy of itself. So we have one linear sequence here, and we should replicate it. And now we have two. And they're exactly the same. Alright, and then last but not least, we have recipient bacteria. That word recipient, it's not defined for us. Is there any way we can do some context clues to kind of figure out what's going on here? Recipient.
Well, there's a donor bacterium and a recipient bacterium. You know, that donor gives, so I'm assuming that the recipient receives.
That perfect use of context clues. A donor bacterium gives away the DNA, whereas the recipient bacterium receives it. You can actually see part of the word receive in that phrase there. Here's the root of that word. Receive. Excellent. Let's take a quick review and make sure we understand what's actually going on with these bacteria. So in a bacterium plasmid, all right, the plasmid is what again about the bacteria? It's what? DNA. All right, so it's a DNA and it's a circle. Now we also know if we take a look at this last paragraph here, that DNA is made up of different genes. Right? Each gene is going to control something. So if we were to take a look at kind of a color version of this, we have 1, 2, 3, again, 4, 5, and they're all exactly the same size. And it's a perfect circle. Anyway, all right, so this whole thing actually becomes one whole plasmid that moves around together that's actually made of 6 different genes. Right? Each gene controls something different. We haven't really covered genetics yet, but like maybe this gene up here is responsible for how fast the bacteria replicates. This gene over here is responsible for making all of the mitochondria inside the bacteria. And every gene has a different job. All right, when bacteria transfer genes to each other, all right, that's part of the way a bacteria works is that it can actually fuse its DNA into another cell. It can cause that cell to then make more bacteria. It's kind of like how things get infected. All right, that bacteria will actually introduce its DNA into you, and they can share DNA with one another. But they can't cut a gene in half. So when they transfer, picture like a little bacteria here, and then we have another little bacteria here. Okay, here's the donor, here's the recipient. What will happen is that the two will actually sort of do a like bacteria mating. Kind of think of it like bacteria mating. One will go next to the other. It kind of extends this two-leg thing. And what will happen is that the DNA, all right, inside of the donor, we would have it's going to be amazing once it works. Wait. Yeah. All right, we have a little miniature version of our DNA in the donor. And eventually the donor is going to want to give the DNA to the recipient. And the way that that happens is we can kind of look at it as a large scale over here, is that they will actually break open the circle at some point. So it'll cut right there. All right, because it'll have to fuse into a line. All right, it can't transmit itself in a circle. So the line breaks open and then it becomes a chain. All right, and the chain then moves from the donor into the recipient, and the recipient kind of takes that DNA in. All right, so you can kind of, it's a little bit like bacterial mating. All right, and that's what these scientists are actually looking at happening. So we see the donor plasmid over here on the side, and the different genes that are located. All right, and the scientists are essentially arguing about how the replication occurs. Right, and each one of these is going to then outline for us how they think it occurs. All right, so student 1 says replication always begins between F and X. X is replicated first, and F is replicated last. Student 2 says replication always begins between F and X. Right, between F and X here. However, the direction of replication varies. All right, what does that word varies mean? Yeah, it depends. It can either go that way, or it can go that way. All right, that's student 2. Student 3 says that replication can begin between any of the two genes, so it can be here, 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 here. All right, and which direction does he say it has to go? Careful, now we're on student 3. Clockwise. It can only go clockwise, which is that way. All right. Last but not least, student 4 says that where can it begin? Anywhere it wants. So it can go here, 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 here. And which direction can it go? Either direction. All right. And then student 1, to finish up what we were talking about, student 1 says it has to begin between F and X, which is here, and it can only go. Ooh, which direction did it go in? How do you know it's clockwise? Let's say it.
Oh, because it starts in X, so X is the first gene to be replicate, and X is the last one. So it starts in X, it goes to the right.
X is perfect. If X is first over here, and F is last, it means it has to go clockwise. Okay. Now that was a lot of information all at once. The tricky part here is trying to balance all of this together. So now there's a couple different strategies when you do this. In real life, you'll have situations like this. We have to analyze 4 different viewpoints. The easiest thing to do is to make something that looks like this. Make some sort of kind of table that outlines the main point of each argument. All right. Now we are going to do that in this case because it's a good skill to have. On the ACT, you won't necessarily have time to build this. You don't want to waste time building it, so you'll have to pull out the information. So we'll actually practice using it. Before we get to the example question, I want you to start to fill out this table. All right, this should be the first thing that you do. So what you see here is for each of the students, you want to list what the first gene would be, which direction it would replicate, and then what the last gene would be, right, based on that information. So take about 2 minutes in your teams right now and fill out this table. You should do the same thing at home. Once you are done filling out this table, you should press play. We'll go through the answers. All right, at this point, if you are pressing play, you've already gone through this table, you are ready to go through the answers. Now, one thing to note on this table is that the way that you write the answers may not have been the same way that we write them together. The formatting is not as important as you understanding the idea of when things get replicated and how things get replicated. All right, so if we start with student 1, student 1 says that the first gene has to be what? X. It has to be X. There's no ors or ifs or ands about this. X is replicated first. That's it. The direction it goes is which direction? Clockwise. Clockwise, and that's what we heard earlier. If it starts at X and ends at F, it has to go in a clockwise direction. And then the last gene has to be F. All right. Now, if we go to the next one, this part really should do that trick because student 2. Student 2 says that it can start at either F or X. Which way will it go? Nice. It can go either way. All right, so how you want to write this is up to you. I think I wrote it as clock or counter. All right, now what we have to see here is that they do tell us where it has to start. All right, this one says it has to start between F and X. All right, now which way it goes is kind of up to whatever the plasmid happens to pick in that case. If F is first, what has to be the last one? X. All right, because it would go F, G, A, S, R, X. So how you want to write this is kind of up to you. I think I would like write it as if F, it has to end at X. What about if it started at X? What would have to be the last one? All right, so if it started at X, it would have to be. Student 3, which gene should be the first gene? Student 3. S, instead? Does it have to be S? And this is the trickiest part of it. Yeah. So here's the thing. This is why this one gets a little confusing. It actually says it can begin anywhere, between any two genes. All right. So our answer here is, I think I wrote it as any. All right. But they give you an example at the end, which kind of makes it a little confusing. It says, thus, if S is first, then A would be second and R would be last. That doesn't mean that it has to go in that direction. They're just giving you an example of if S were first, then A would be second and R would be last. It doesn't mean it has to go that way. All right, you can tell that because it says if, not it has to. All right, and then last but not, oh wait. So then if it ends at any, how could you be really specific with the last gene? It would have to be the gene to which direction of the first one? Nice. We know that this one's going clockwise, which means the last gene has to be one counterclockwise from whenever the first one is. Meaning that if you started at G, it would go G, F, X, R, S, A, and so A would have to be the last one. All right, so how you write that again is kind of up to you. So like if we started at S, it would go S, A, G, F, X, R, and R would be the last one, which is like one just the counterclockwise. So I don't know how you really want to write it, but I think you can write it as one counter first. Clear way to write it. Last but not least, student 4. All right, that part shouldn't be that tricky. Where did it say they start? Anywhere. Which direction can it go? Any direction. So now, how would you write last gene if you want to be real specific? All right, because I think originally in the answer sheet I wrote any here, and maybe any isn't the best answer. Because if you started at G, for example, which two could be the last one? Yeah, it would either go start at G and go FX R. So it would go FX R S and end at A. Or if you started at G, it could go G A S R X F, and F would be the last one. So I don't know. I guess you could really write like adjacent here because it would have to be adjacent to the first one, either to the left or to the right, but it could be either one. Again, this is like kind of confusing, but it's just the idea that you understand about how it plays out. All right, once you have this done, now you can definitely answer a question like the example question, which is pretty straightforward. All right, they're arguing about the order in which they're replicated. All right, that was a pretty straightforward question as long as you go through and read through the paragraph. It's also got an explanation at the bottom if you're confused about that. In order to really get into the meat of these, what we're now going to use is the sample table that we already made to answer questions 2, 3, 4, and 5. Right, what you want to do in your groups or at home if you're watching this video is answer questions 2, 3, 4, and 5. As you go through these, make sure that you're really understanding which direction the plasma is moving in. When you're ready to go through these answers, press play, and we'll go through them together. Right now, you should press pause. All right, at this point, if you're pressing play, that means you're ready to go through questions 2, 3, and 4. And even 5. One thing to all these questions though, that you want to make sure that you're noting is the difference between the words could and must. What you see here is that a few of your scientists actually give you options. So you notice like for scientist or for student 2, she gives you the option of is it F or is it X? And she also says that it could be clockwise or counterclockwise. You want to make sure that you are noting that because 2 kind of give you some options, where student 1, on the other hand, is really specific. It says that first gene has to be X. So student 1 is a lot more about musts, like it must start at X, it must go clockwise, and it must end at that. Where some of your other scientists give you some more options in terms of the word could. So if we take a look at question 2, it says, which students would agree that replication could end at gene G? Now, not must end at gene G, but could end at gene G. Who would say that? Excellent. The only 2 that give you that probability, because we have the words any here. All right. 3 says it can end anywhere, and 4 says it can end anywhere. Right? So what you want to do here is recognize that both of these, they cannot end at G. The only 2 that can or could end at G are 3 and 4. If we take a look at number 3, the model of student 1 differs from the model of student 3. And that student 1 says, so now you have to be really careful about this question because that gives you a couple of things. You're trying to figure out not only the differences between 1 and 3, but you're looking for something that student 1 does say that student 3 goes against. All right, so you're not just looking for something that student 3 doesn't say, but you're looking for something that student 1 does say that student 3 doesn't say. All right, again, you get kind of confusing if you start talking about these model specifics. So, does student 1 say that it goes clockwise? Yeah.
Yes.
Yep. But it's not the answer because who else says it goes clockwise? Not student 3. So in this case, that can't be the answer because that doesn't make them different. All right. What about the next one? Replication proceeds counterclockwise. First off, does student 1 say it? No. So we already know that that's not the answer. Next one. Gene X must be the first gene replicated. And now we see the word must here. All right. So X. Does student 1 say that X must be first?
Yes.
Yes. Does student 3 disagree with that? Yes. All right, now you'll notice, student 3 says that could it start at X? Yes, but does it have to start at X? No. And that's why this is the right answer. All right. Gene S could be the first gene. Student 1 would disagree with that. Right? All right, they have to start at X. All right, so let's go on to number 4. Supporters of all 4 models would agree that, and you'll notice in all of these are all coulds, not musts. All right, they're all coulds. So a plasmid, now the easiest way to take a look at this is you're trying to, like you could go through and test every single one, see which one is right and which one is wrong. Another way to take a look at this question is to find the pickiest student. Which student do you think is the pickiest student of all the students? Why do you say 1? Nice, fantastic. All right, student 1 is very specific about what she says has to happen inside of this. There's no ors, there's no ands, there's no possibilities. So the idea is that if we can get 1 to agree with it, we can probably make the other ones agree with it as well. Oh, so which one is it? Which is the only one of these that 1 would agree with? Nice. All right, and you can tell that right away. Do you even need to look in the middle? No, but I'll tell you this, you should. Because on the actual ACT, there were 2 options. There was another option that said X, S, R, A, F. It started with X, and it ended with F. But what's the problem with the middle?
It skips one, so
Yeah. If you were to go back, let's see, we go X, R, A, S, F would be totally out of order. Yeah, so you just got to be careful because that question or something close to that was actually on the ACT. All right, in this case, repeat. All right. Yeah. All right. Let's take a look at 5. Student 2 and student 4 would agree that, and you'll notice you have 2 different options. You have 3 of these cans, so those are the could categories. So that could happen, and 1 must category. All right. Which of these would student 2 and 4 both agree on? What do you think? I see. Nice. All right. In a question like this, with a nice table like this, it's easy. You just try to look for, is there anything the same? All right. Clockwise and counterclockwise, clockwise and counterclockwise. So C would be there. Now this is nice when you have a table. However, on the ACT, you're not going to have time to make this for a problem. All right. This table would take you probably 2 or 3 minutes to make, which doesn't seem like a long time, but that's almost an entire passage worth of time. So what I want you to do for the rest of these, I want you to take the top of your paper and like fold it down so that you can't see that table anymore. All right. So that for the next ones, for 6, 7, and 8, you are only using the scientist's description and the diagram instead of using the table. All right. Try not to make yourself one-dimensional, only relying on the table. So try 6, 7, and 8 just based on the passage and the diagram instead of using the table. All right. If you're watching at home, press pause now, and you want to go through questions 6, 7, and 8. When you are ready to go through the answers, press play. At this point, if you are pressing play, you're ready to go through the answers for questions 6, 7, and 8. Remember, without cheating using the table, only using the passage and the diagram. Let's take a look at 6. Which students would agree that gene S must be the third gene transferred? All right, now the ACT will not be nice and bold and underline words like this. You have to be able to start pinpointing these. That S must be the third gene transferred. Which is the only student that would say that? Student 1. All right, now the tricky part of this is a lot of people automatically go for D. Because if you look just a few questions ago, we had this order where S could be the third one, and they all agree with that, is what this question told us. But the tricky thing you have to see here is that we were talking about coulds in this case, not musts. All right, student 1 says it must go X, R, S, A, G, F. That's the real picky one, so S has to be third. But if you even look at just student 2, for example, they could start at X, so it would go X, R, S, and S would be third. But the student also says that you could start, instead of at X, you could start where? At F, and go the other way. In that case, A would be the third one, not S. So this is a case where could is different than must. So in this one is like, what is the only student that says that this has to be the way it goes, not that this could be the way it goes. And that would only be student 1. Everybody else to loosey-goosey theories to say that would have to happen. Number 7, which students believe that any of the 6 genes on the plasmid could be the first gene transfer? Now we have a could situation. All right, who says that any of them could be the first one?
3 and 4.
Max, 3 and 4. All right, they say it can begin between any 2 genes, can begin between any 2 genes. All right, this one right here is too restricted. It says it has to be between F and X, and this one says it has to be X first. That means that 3 and 4 is the right answer. Last but not least, number 8. Which of the following phrases best describes the major point of difference between the model of 3 and 4? What's the biggest difference between 3 and 4? Now, with that table, this question would be easy. But you don't have the table. So where do you look?
I said it would be the replication direction.
Nice. All right. In 3, so the answer was the replication direction, which is B. And you'll see that here, it has to go clockwise. And here, you'll see that it can proceed in either direction. Everything else is the same. We're going to start with it. Remember, don't get tripped up with this thus business. All right? This is just an example of what could happen, not what has to happen. All right. At this point, it's quiz time. You feel like you have all of these questions down and you are ready to go. All right, take the quiz. If you are at all uncomfortable with these, you want to make sure that you rewind. Go to the part that you're confused about and watch the video. Good luck.